E. coli, here's the phage. You'll get them from this box. You need 1 E. coli and 1 phage per team, if I have enough.
Phage is the dark one?
The dark one is E. coli. And you're going to need 8 of these. I don't know if I'm going to have enough for teams, for that many teams.
I'm with them. I'm with them.
No, I have 5, 6, 7 teams. I have just enough, I think. I don't know. We'll see what happens. Okay, so then these little tubes you're going to get out and you're going to have to mark it with your Sharpie pen. See how it has number 1?
And you said how many of those?
8.
8.
So I got 1, 2, 3, 4, 5, 6, 7, 8, which represents 10 to the negative 1, 10 to the negative 2, 10 to the negative 3. 10 to the, that's what it represents. But that was too much to write on here, so we just put 1, 2, 3, 4, 5, 6, 7, 8. Got it? Okay.
Yep.
So you're going to need your rack, your E. coli, your phage, and 8 empty tubes that you're going to label. Okay. First thing that you do, in blue, ignore the green, it doesn't exist right now. The blue. Do this entire step first. So, first thing you have to do is put 900 microliters of phosphate buffered saline solution into these tubes. So you're going to use your big pipette. It's with the big tips in the blue box. You're going to set it to 090, that's 900 microliters, and you're going to squirt, you can use the same tip. Squirt phosphate buffer into each of these tubes. 900 microliters into each of those little tubes. Then you're done with your big pipette. Now you're going to use your little pipette, and you're going to take, first thump this to mix it. You know, thump it, make sure it's closed, thump it. Mix your phage stock. Transfer 100 microliters into the first tube. Thump it to mix it. Get a new tip. Transfer 100 microliters into the second tube. Thump it to mix it. Change your tip. Transfer 100 microliters to the next one. Thump it, change your tip, and so on down the line. So each time you're diluting it by 1 decimal place. You're diluting it by a factor of 10. Okay? That's your serial dilution. It's an easy one, because each time we're transferring the same amount, each time we're diluting it by the same factor of 10. So, once you have all your tubes ready, you're actually going to work with the higher numbered ones. Well, what's going on here? I give you a little cuvette of phage stock. There's phages in this solution. You have no idea how many. Maybe this would be, I took maybe a milliliter of sewage and I made it into a solution. And then I dispensed it into these little cuvettes. What I want to know is, what is the density of phages in the sewage? Well, when I made this, I had no idea. I still have no idea. What I'm going to do is, there's too many in here probably to count, I'm guessing. So I'm going to dilute it down. And then I'm going to grow it. So I have a small enough number that I can count them, but not so small that it's an insignificant number. I have no idea what that's going to be. So I'm going to make a range. And then, once I get my information how many grew here, I'm going to multiply back and figure out how many were in my sample. So the whole purpose of doing the serial dilution is to try to calculate how many you had in your original stock solution. But because there's probably too many in there for me to just plate them and count them, I need to dilute them down to a more manageable number. But I have no idea what that number is, so I'm going to have to make a range. Okay, so I've done my serial dilution, and now I'm guessing that it's going to be in that range where I'm going to get a low enough number that I can count them, but enough that it's a significant number. So now you're going to transfer 100 microliters from this into your agar tube. What's in your agar tube? Agar with E. coli in it. So So the agar is there for the bacteria to grow. The bacteria are there for the viruses to grow, the phages to grow. So I gotta get all three in there together. All right? So, do the entire first step first and let it sit for a minute while you take care of this. You gotta get 4 top agar tubes, and they're supposed to be molten. And then you let them cool to the point where they're still molten, but they're not too hot for the bacteria and the phages. And then you use a pipette to transfer 100 microliters with your small pipette 100 into each of these. So you can use the same tip for that. You're going to transfer 100 microliters in that one, 100 microliters in that one, 100 microliters in that one, 100 microliters in that one. Technically, you should probably use a new tip each time, but I don't think it's going to hurt it too much if you use the same tip as you go. But, you know, the first time the tip is clean, the second time you're adding E. coli, there's some lingering in from the first time. So it could have a little bit of an effect. But okay, I got my E. coli in my top agar. Now I have to add my viruses. So I'm going to have to use a new tip to transfer this solution, 100 microliters, into that. This one into that, this one into that, and this one into that. Okay? So you're going to have to label your tubes also, 7, 8, 9, 10. Okay? Then you're going to vortex them, and hopefully you've worked fast enough that they're still in a molten state, so you can pour them onto the Petri plates. Now the Petri plates are called half full. See, it's half full. So that means it's got a solid agar base. You're simply going to open your plate a little bit, pour in the agar from your test tube, all of it, close it, and then swirl gently to spread it around the top of your plate. This is the pour plate technique. We put our bacteria into the agar, and we poured it on top. Now the bacteria are going to form what we call a bacterial lawn. We literally seeded the agar with E. coli. And when we pour it onto the surface of the base agar and let it solidify, the bacteria are going to grow on the surface in a lawn. So all the agar is going to become cloudy as the bacteria grow in it. Do you remember on an earlier lab quiz you had a test question about what would happen if I grew my facultative anaerobes in the agar as opposed to on the surface of the agar? Remember that? That's what's going to happen here. You're doing a pour plate method. So some of the bacteria are going to be in the agar, and some of them are going to be more on the surface of the agar. Which ones are going to be bigger? The ones on the surface, because they're facultative anaerobes, so they can grow on the agar where there's air, and they can also grow in the agar where there's not so much air. But if you do your metabolism aerobically, you get 32 ATP per molecule of glucose. If you do it anaerobically, you only get 2. So the ones on the surface that get air are going to have more energy, and they're going to form bigger colonies. The ones that are in the agar can survive because they're facultative anaerobes, but they're only going to get 2 ATP per molecule of glucose, so they're not going to have as much energy, so they're not going to have as big a biomass, so the colonies are going to be small. And on your Lab Quiz 5, you're going to have to recognize pictures. Yes, there will be pictures on your Lab Quiz 5. You'll have to recognize and identify pictures of pour plates, as opposed to pictures of spread plates, as opposed to pictures of streak plates, Kirby-Bauer plates with antibiotic discs and zones of inhibition. So you have to recognize different types of plates. Okay, if you do the pour plate method, you're going to add your E. coli to your agar, and then you're going to add your viruses to the agar, because the viruses have to grow in the bacteria. So if you have a lawn of bacteria and there's viruses in there, the virus is going to infect the first bacterial cell, be replicated, the new virions are going